Welcome to the deep dive. Today, we're going to be exploring a study that kind of sits right at the intersection of chemistry and botany, and also a potential new treatment for gout.
Oh, absolutely. It's always exciting to see how scientific research can uncover hidden potential in natural resources,
Exactly.
especially when those resources have a long history of traditional use.
Yeah, so let's dive into the source material for this deep dive.
Yeah.
We're focusing on a 2004 study
All right.
that was published in the Journal of Separation Science.
Okay.
The title is long, um, Using Analytical Multidimensional Isocratic HPLC Methods of Separation to Isolate Active Constituents in Natural Products.
Wow, quite a detailed title.
Yeah.
But it clearly outlines the focus of this research,
Yeah.
isolating specific components from a complex natural source.
Right, and the source they're looking at is Clerodendrum floribundum, an Australian native shrub.
Okay.
Before we get into the specifics of the study, can you give us a little background on this plant and why it caught the attention of scientists?
Of course, Clerodendrum floribundum has a long history of medicinal use by Aboriginal communities in Australia.
Interesting.
It's been traditionally used to treat a variety of ailments, including inflammation, infections, and pain.
Okay.
This traditional knowledge piqued the interest of researchers who suspected the plant might contain bioactive compounds with therapeutic potential.
Interesting. And they were specifically interested in its potential application for gout treatment, correct?
That's right. Gout is a painful inflammatory condition caused by a buildup of uric acid in the joints.
Okay.
Scientists discovered two compounds within Clerodendrum floribundum named CF1 and CF2 that showed the ability to inhibit xanthine oxidase,
Okay.
the enzyme responsible for uric acid production.
So by inhibiting xanthine oxidase, these compounds could potentially reduce uric acid levels and alleviate gout symptoms.
Exactly.
That's a pretty exciting prospect.
Indeed. However, isolating and purifying these compounds from the complex mixture of chemicals present in the plant is a significant challenge. That's where the sophisticated separation technique used in this study comes in.
Okay.
Multidimensional, high-performance, liquid chromatography, or MD-HPLC.
Okay, let's unpack MD-HPLC. It sounds pretty complicated. Can you break it down for us?
Certainly, imagine a highly efficient sorting system.
Yeah.
But for molecules,
Okay.
MD-HPLC essentially uses two different separation stages
Okay.
to isolate specific compounds from a complex mixture.
Okay.
Each stage uses a different type of column with unique properties.
Right.
So you have two separate opportunities to discriminate between compounds based on their chemical characteristics.
It sounds like a much more precise way to isolate specific compounds compared to traditional single-column HPLC.
Yes, definitely.
What makes this particular study unique in their application of MD-HPLC?
Well, in this case, the researchers chose to use isocratic elution for both stages of the MD-HPLC process.
Okay.
Isocratic elution means that the composition of the solvent or mobile phase remains constant throughout the separation.
Why is that significant?
Using a constant solvent composition simplifies the process and speeds things up,
Okay.
because there is no need for reequilibration between different solvent mixtures, which is required in gradient elution.
Right.
In gradient elution, the composition of the mobile phase changes over time,
Okay.
which can lead to better separation in some cases,
Yeah.
but it also adds complexity and time to the process.
So they prioritize speed and efficiency, which makes sense given the goal of isolating and characterizing these compounds.
Yes.
But wouldn't a constant solvent composition limit their ability to separate a diverse range of compounds?
That's a valid point, but the researchers employed another clever technique to enhance separation: heart cutting.
Heart cutting, what does that involve?
Heart cutting involves selectively transferring a specific portion of the mixture flowing out of the first column to the second column.
Go ahead.
It's like taking a snapshot of a specific moment in the separation process where you know your target compound is likely to be present. This selected portion, or heart cut, is then further separated on the second column with different properties.
They were basically fine-tuning the separation process
Yes.
by focusing on a very specific window of the initial separation. That sounds meticulous.
It is, and the researchers went to great lengths to optimize this heart cutting technique. They carefully tested different heart cut fractions and volumes to determine the optimal conditions for isolating CF1 and CF2 with the highest purity.
It seems like achieving high purity is a crucial aspect of this research.
I
Why
feel-
that's so important?
Purity is paramount, especially when you're considering potential medical applications.
Right.
Any impurities could have unintended and potentially harmful effects.
Of course.
Having a highly pure form of CF1 and CF2 ensures that any observed effects are truly due to these compounds and not some unknown contaminant.
It's like making sure you are using the correct ingredients in a recipe.
Exactly.
You don't want to accidentally add something that could spoil the entire dish.
Right.
So how successful were they in isolating these compounds?
That's where things get really interesting. Despite the challenges, the researchers managed to obtain CF1 and CF2 with remarkable purity.
Wow.
Approximately 95% for both compounds.
95% purity. It's impressive.
It is.
It sounds like they were quite successful in developing a method for isolating these potentially therapeutic compounds.
Yes.
But I imagine there were still some challenges along the way.
There were indeed. Obtaining a high level of purity is just one piece of the puzzle.
Right.
Another challenge they faced was maximizing the recovery rate of CF1 and CF2.
Okay.
In other words, how much of the compound could they actually extract from the plant material?
That makes sense. It's one thing to isolate a tiny amount for research purposes, but scaling up production for widespread medical use is a whole different ballgame.
Exactly. And that's where we'll pick up our discussion in the next part of this deep dive. We'll delve into the specific challenges they encountered with compound recovery and what those challenges mean for the potential development of a gout treatment based on these compounds.
Sounds fascinating. I'm eager to hear more about the details of their separation process and the results they obtained. I think our listeners will be too. Stay tuned, everyone.
Welcome back to the deep dive. Before we delve into the specifics of the separation process and the results obtained, let's quickly recap the key aspects we discussed earlier. We are examining a study by Victor Wong, Alan P. Sweeney, and R. Andrew Shalliker, published in the Journal of Separation Science in 2004.
Okay.
The study focuses on using multidimensional isocratic high-performance liquid chromatography, or MD-HPLC, to isolate two potentially therapeutic compounds, CF1 and CF2, from the Australian native shrub Clerodendrum floribundum.
That's right. We learned that this plant has a long history of medicinal use by Aboriginal communities, and these particular compounds have shown promise in inhibiting xanthine oxidase, which plays a key role in uric acid production. Inhibiting this enzyme could potentially offer a new avenue for treating gout.
Precisely. Now let's get into the details of how the researchers went about separating and purifying these compounds. As we mentioned earlier, they employed a two-dimensional HPLC system with isocratic elution.
Right.
What's interesting is their choice of stationary phases for each dimension. For the first dimension, they used a nitrile column, which separates compounds based on their polarity and interaction with the nitrile functional groups.
Okay.
This initial separation helps to simplify the complex plant extract and differentiate compounds based on their affinity for the nitrile stationary phase.
So the nitrile column acts as a first filter, so to speak.
Yes.
Creating a preliminary separation of the compounds in the plant extract.
Yeah.
What about the second dimension? What type of column did they use there?
For the second dimension, they used a C18 column, a very common and versatile stationary phase in HPLC.
Okay.
C18 columns are packed with silica particles that have been chemically modified with long hydrocarbon chains.
Right.
This creates a nonpolar environment, allowing for separation based on the hydrophobic interactions between the compounds in the stationary phase.
Okay.
Compounds that are more hydrophobic will interact more strongly with the C18 stationary phase and elute later. Okay.
While more hydrophilic compounds will elute earlier.
So the C18 column adds another layer of separation, further discriminating between the compounds that were initially separated by the nitrile column.
Exactly.
This two-dimensional approach seems quite powerful in achieving high resolution and purity.
Indeed. But the key to successfully utilizing this two-dimensional system lies in the heart-cutting technique we touched upon earlier.
Right.
The researchers didn't simply transfer the entire eluent from the first dimension to the second.
Okay.
They carefully selected specific fractions to transfer based on the time at which they eluted from the first column.
Okay.
Remember, they were looking for two specific compounds, CF1 and CF2, which they expected to elute at particular time intervals.
It's like they were carefully snipping out specific sections of a continuous thread.
Yes.
Choosing only the segments where they knew their target compounds were most likely to be present.
That's an excellent analogy.
Okay.
They essentially fine-tuned the separation process by focusing on those specific heart-cut fractions.
They experimented with different heart-cut volumes and time intervals, meticulously optimizing the transfer process to achieve the highest purity possible for CF1 and CF2.
Right.
They found that the optimal heart cut window for CF1 was between 2.10 and 2.3 minutes,
Okay.
while for CF2 it was between 2.2 and 2.40 minutes.
So even though these compounds might have eluted relatively close to each other in the first dimension, they were able to differentiate them more clearly in the second dimension by carefully selecting the appropriate heart cut fractions.
Exactly.
This highlights the level of precision and control that MD-HPLC offers.
Absolutely. And the results speak for themselves.
Right.
As we mentioned earlier, they achieved a remarkable purity of approximately 95% for both CF1 and CF2.
Wow.
This level of purity is crucial for any future studies investigating the biological activity and potential therapeutic applications of these compounds.
It certainly is a testament to the power of MD-HPLC and the meticulous optimization work done by the researchers.
Absolutely.
But as you hinted earlier, achieving high purity is only one part of the equation.
Yes.
They also encountered challenges with the recovery rate of these compounds. Can you elaborate on that aspect?
Yes, of course. While they were able to achieve exceptional purity for CF1 and CF2, the amount of each compound they were able to recover from the plant extract was relatively low.
Oh, okay.
In their best case scenario, they recovered only about 41% of CF1 and 58% of CF2.
That's a significant loss.
Yeah.
What factors might have contributed to this low recovery rate?
Several factors can influence sample recovery in chromatography. One possibility is that some of the compounds might have irreversibly bound to the stationary phases in the columns,
Okay.
even with the addition of phosphoric acid, which is often used to improve recovery.
But
Another possibility is that some material might have been lost during the transfer process between the two dimensions,
right.
especially during the delicate heart cutting step.
I see. It's like trying to carefully transfer a small amount of a precious liquid from one container to another.
Exactly.
There's always a chance of losing some drops along the way.
Exactly. And when dealing with small quantities of potentially valuable compounds, even small losses can have significant implications,
Right.
especially when considering scaling up production for potential pharmaceutical applications.
That brings us to a crucial question. What are the implications of this low recovery rate for the future development of CF1 and CF2 as a potential gout treatment?
That's a critical point, and one that the researchers themselves acknowledge in their study. While they successfully demonstrated a method for isolating and purifying these compounds with high purity,
Right.
the low recovery rates pose a significant challenge for scaling up production to a level that would be needed for clinical trials and eventual pharmaceutical development.
So while this study represents a significant step forward in understanding the potential of Clerodendrum floribundum and its bioactive compounds, the journey towards a new gout treatment is far from over.
Indeed, further research will be needed to optimize the recovery rates of CF1 and CF2. This might involve exploring alternative stationary phases, modifying the mobile phase composition, or refining the heart cutting technique.
Okay.
It's a reminder that scientific progress is often an iterative process, building upon previous discoveries and refining techniques to overcome new challenges.
Absolutely. It's a testament to the perseverance and ingenuity of scientists in their quest to uncover nature's secrets and harness its potential for the betterment of human health. And speaking of uncovering nature's secrets in the next part of our deep dive, we'll discuss the broader implications of this study and explore what the future might hold for CF1 and CF2 in the fight against gout. Stay tuned. Welcome back to the deep dive. We've been on quite a journey exploring the potential of Clerodendrum floribundum as a source for a new gout treatment. We've learned about the plant's traditional uses, the exciting discovery of the bioactive compounds, CF1 and CF2, and the sophisticated techniques used to isolate them.
Yes, we've delved into the intricacies of multidimensional high-performance liquid chromatography, or MD-HPLC, and how the researchers were able to achieve remarkable purity in isolating these compounds.
But we've also discussed the significant challenge they faced, the relatively low recovery rate of CF1 and CF2. This limitation raises some important questions about the feasibility of scaling up production for clinical trials and eventual pharmaceutical development.
Indeed, and that leads us to a broader question. What are the implications of this study for the future of drug discovery, particularly in the context of exploring natural resources for potential therapeutic applications?
That's a great point. It seems like this study highlights both the immense potential and the inherent challenges of looking to nature for new medicines.
Absolutely. This research beautifully illustrates how traditional knowledge can serve as a springboard for scientific discovery for centuries. Aboriginal communities have relied on Clerodendrum floribundum for its medicinal properties, and now modern science is beginning to unravel the chemical basis for those traditional uses.
It's a fascinating example of how ancient wisdom can guide cutting-edge research, leading to the identification of promising new compounds like CF1 and CF2.
It certainly underscores the importance of exploring and preserving traditional knowledge systems, not just for their cultural value, but also for their potential contribution to modern medicine.
So in a way, this study represents a bridge between ancient wisdom and modern scientific techniques, highlighting the potential synergy between these seemingly disparate worlds.
Precisely. And it's a reminder that nature holds a vast reservoir of potentially therapeutic compounds waiting to be discovered and explored. But as this study also demonstrates, the path from identifying promising compounds to developing a safe and effective treatment is often long and challenging.
Right? It's not enough to simply isolate a compound in a lab. We need to be able to produce it in sufficient quantities and ensure its safety and efficacy through rigorous testing.
Exactly. And in the case of CF1 and CF2, the low recovery rates pose a significant hurdle that needs to be addressed before we can even begin to think about large-scale production and clinical trials.
So while this study lays a solid foundation for further research, there's still a long road ahead before we can say for certain whether CF1 and CF2 will fulfill their promise as a potential treatment for gout.
That's true. However, even if these specific compounds don't ultimately make it to the market, the insights gained from this study will undoubtedly inform future research in this field. It highlights the potential of MD-HPLC as a powerful tool for isolating and purifying natural compounds, and emphasizes the need for ongoing innovation in separation and purification techniques.
It also reminds us of the importance of perseverance in scientific research. Setbacks and challenges are inevitable, but they often pave the way for new discoveries and advancements.
Absolutely. And who knows, perhaps future research will uncover even more potent compounds within Clerodendrum floribundum or other plants with a history of traditional use in treating gout.
It's an exciting prospect to consider the world of natural products. Research is full of untapped potential, and studies like this one inspire us to keep exploring and innovating in our quest to improve human health.
Well said. It's a testament to the power of curiosity, collaboration, and the relentless pursuit of knowledge that drives scientific progress.
On that note, I think we've reached the end of our deep dive for today. It's been a fascinating journey exploring the intersection of chemistry, botany, and traditional knowledge in the search for new treatments for gout.
It's always a pleasure to delve into these complex and intriguing topics.
And to our listeners, we hope you've enjoyed this exploration into the world of Clerodendrum floribundum and the potential it holds for treating gout. It's a story that highlights the interconnectedness of nature, traditional knowledge, and modern science, and the incredible possibilities that emerge when we bring those worlds together. Until next time, keep diving deep and keep those brains buzzing.